Hello, my name is Diana Ibarra, and I'm a first-year graduate student in Dr. Peng's lab. We are interested in studying pain and its underlying mechanisms. Our lab uses animal models, specifically rats, and neurosignal recordings in order to do so. Pain is a complex sensory and emotional experience that is widely misunderstood. It is crucial not only to understand its fundamental role, but understanding will also allow us to improve quality of life. Today, I will be presenting effect of graded peripheral electrical stimulation on brain activity. The goal of the study was to demonstrate the use of graded electrical stimulation and its effects on the activation of four specific brain areas consisting of the anterior cingulate cortex, the left and right amygdala, and the ventral tegmental area, which are all associated with pain. We used a total of 7 Sprong dolly rats in the study. We drilled holes in the four targeted areas and intracranially implanted electrodes. We then secured these electrodes with dental cement. After the cement dried, we attached a wireless recording device along with a dongle which connects to the computer to transmit the brain activity in a visual manner. There were two experimental conditions within this study. The first condition, which was pre-formalin, tested the parameters of electrical stimulation. We used three different frequencies that consisted of 1, 50, and 100 Hz, as well as four different amplitudes in terms of 5, 10, 50, and 100 volts. The pause width duration was set at 1 millisecond, and the electrical stimulation was given for 11 seconds. The interstimulus interval was a total of 3 minutes. These same parameters were used in the post-formalin condition. Formalin was injected in the left hind paw to induce an acute inflammatory response. This data was then analyzed using MATLAB. Figure 1 illustrates raw waveforms and power spectrums of the ACC region in the pre-formalin condition when electrical stimulation is being administered. We can see the 1, 50, and 100 Hz in conjunction with the voltages given. As you can see here, 300 seconds translates to 5 minutes, which is when the 5 volts was given at 1 Hz. We can see that this electrical stimulation produces little to no change. The power seems to remain stable. However, if we take a look at 10 volts given at 100 Hz, we can see a brief suppression followed by a rebound phase. If we take a look at Figure 2, this displays analyzed 10-second data in the ACC region. For the pre- and post-formalin, you can see a slight trend in the pre-formalin condition. As Hertz and voltage increases, so does the brain activity. For post-formalin, we can see an increase in power when formalin was injected. However, this activity then remains constant, which can possibly be attributed to the administration of the electrical stimulation. This pattern can be seen in the figures for the right amygdala as well. Overall, these findings suggest that in the pre-formalin condition, though lower the electrical stimulation intensity, no significant change in power was observed. However, increasing intensity produces a brief inhibition followed by a rebound effect. There is an increase in power after the injection of formalin in the post-formalin condition, but the activity seems to remain stable, possibly due to the administration of electrical stimulation. We did not see the anticipated increase of power when delivering electrical stimulation in both the pre- and post-formalin conditions. We will modify the current parameters for future studies. Thank you so much for listening.